Hi there, welcome back to our series on the cell. So last day, remember, we took a look at the vacuoles, and today we're going to look at plastids. So just to remind you, we're not really going to focus on all the little nitty-bitty bits of the cell. We're going to focus on the parts that actually help to define a plant cell and distinguish it from animal cells. So we took a look at the vacuole. Here's our vacuole here, nice and fluid-filled, lots of important functions. And we're going to take a look at the plastids today. One kind is a chloroplast, you can see here and here behind the vacuole. And next time we're going to take a look at the cell wall. Okay, here we go into the world of plastids. So here we can see chloroplast within the cell. It's green, it's photosynthesizing. These little white things are starch grains here in this illustration. We also can take a look at cells using transmission electron microscopy, and that's what this micrograph is here. And here are chloroplasts right there. There's our vacuole. Okay, so basically, our plastids are organelles. They've got double membranes and they contain DNA. They also have compartments in them, and so in the case of the chloroplasts, they have thylakoids you might be familiar with. And what's also interesting about them is they divide independently of the rest of the cell. So that's kind of interesting. Now, another really interesting thing, I think, is that way, way back, they kind of arose because there was a predatory protozoan that engulfed a photosynthetic bacteria. And that photosynthetic bacteria later became the chloroplast. Okay, so we had our protozoan becoming this photosynthetic organism, and we can see that they actually diverged into two groups of algae, the green algae and the red algae. And it was the green algae that actually gave rise to the land plants. So yay, green algae. What I also find really interesting is that even though that little photosynthetic bacteria gave rise to the chloroplast, throughout plant evolution, these plastids became really important for all sorts of different functions. They not only function in photosynthesis, but they also make pigments, amino acids, and lipids. They also are responsible for the storage of lipids, carbohydrates, and proteins. They can sense gravity in some cases. They regulate the stomata and lots of different kinds of functions. And in fact, we don't really know all of them. This is an illustration from your textbook showing you that the precursor to all the plastid types are proplastids. So these proplastids can differentiate. Here's this differentiation into the chloroplast. Can also differentiate into a chromoplast. But a chromoplast can differentiate into chloroplast. A chloroplast can differentiate into a chromoplast. The proplastid can differentiate into an amyloplast, and this can differentiate into a chromoplast. So lots of flexibility with these plastids. Well, I actually like this illustration a little bit better. And basically it's showing you this same idea, this proplastid differentiating. And in a lot of sort of cells that are getting ready to become photosynthetic, maybe like an embryo that hasn't yet met the sunshine, we have this thing called an idioplast, so that it's ready to go when light hits. And so we get this chloroplast, which is our photosynthetic plastid. We also get the chromoplast, which contains things like carotenoids and xanthophylls, pigments that are important for fruit ripening, for petal colors in flowers, also for UV protection. When we have leucoplasts, which are colorless, the most common one that we'll see in the lab is the amyloplast, which contains starch and stores starch. There are other ones as well that store protein, proteinoplasts, and we have ones that store oil as well, eleoplasts. Now, proteins and oils are stored in other places in the cell as well, but they are stored in plastids. And so these are the plastids that I would expect you to know, but there are many other ones, and I'm just going to show you a few. Drought-resistant or drought-tolerant plants. So it is where the chloroplast can go dormant for a little while, and then when conditions are right, can quickly develop back into a chloroplast. Another interesting type is a tannosome-forming plastid that can come from the chloroplast. and it's where the chloroplast membrane forms pockets that fill with tannins. So basically it's responsible for synthesizing, producing, and condensing tannins and other kind of phenols, and probably provides defense against herbivores and pathogens, and perhaps even protection against UV radiation. When chloroplasts start to degenerate, they're called gerontoplasts, and it's kind of happens during senescence of plant foliage. Generally seen as a process where the grana of the thylakoids become unstacked. We're going to talk more about thylakoids when we talk about photosynthesis. Some recently researched types of plastids are kinds of leucoplasts, so these colorless plastids that include things like the phyloplasts. And the phyloplasts appear, they're quite a new type of plastid to be important in the synthesis of cell wall precursors. So actually really central to the evolution of vascular plants. Phenyloplasts, they actually are storage areas for phenolic secondary metabolites, and they've just started to be researched, and I think most of the research has happened in vanillabine. The phenolic, vanillin is actually phenolic secondary metabolite. So kind of interesting group, but remember you really only have a small number that you need to know about. Okay, so here is a cell. This is a cell from a bean actually, and the cell contains many amyloplasts. So the amyloplasts stain purple here because we use a stain called IKI, or potassium iodine, which stains starch purple, and if you leave it on a long time it'll stain it black. Here we have a cell of a red pepper. So the outer flesh of the red pepper is red when it's mature. So part of fruit ripening often is changing color into something that looks yummy to a animal, usually a mammal. And here we have the little chromoplasts, and then we have our central vacuole in the middle. Here's a TEM, transmission electron micrograph, of a chloroplast, and you can see it's got its thylakoids all stacked up, but you can also see it has other things too. It has some oil bodies in it. You may even find starch grains in chloroplasts. So many of these plastids can contain more than one compound or substance. Here is a cell that's an example of one that contains more than one kind of plastid. So here we have some plastids here. I think they're a myeloplast, but we're going to test it out by using IKI in a minute. And then we have our chromoplasts. So this is actually taken from what we call a yam from the grocery store, but really, to be honest with you, the grocery store yam is actually just orange variety of a sweet potato. I hate to break it to you. But if we stain it with IKI, look how lovely that looks, right? Here we have a lovely cell here, and you can see the amyloplasts with the chromoplasts. So nice combo of purple and orange. So these slides are actually made from students in Biology 210. So that's pretty neat. We also have changes in colors due to the seasons, right? We have autumn coloration. Now, at the end of a season, often plants senesce, and that's the changes that occur when an organism or part of an organism is mature. So we can have leaves falling off at the end of the season. We can also have fruit falling off after it's mature, and this process is called abscission, the dropping off of plant organs. Now, leaves, it's often accompanied with changes of color, and so we have many leaves exhibiting autumn coloration, yellow, orange, reds, and purples before they fall off. During this process, chlorophylls are broken down and the underlying pigments are revealed. So that's kind of neat, right? So we have this degradation. That used to be what people thought was the reason why we have the autumn colors, was because of this reveal, the big reveal of all of these other pigments that are in the chloroplasts. But actually, as it turns out, more pigments are produced in some plants, right? So we have the production of these anthocyanins. Now what's kind of interesting though is that not all plants change leaf color during senescence. So is there actually an advantage? Why do they do that?
So what is the adaptive advantage? Well, until recently it had been thought that the consequence of chlorophyll degradation resulted in the exposure of these carotenoids, xanthophylls, and anthocyanins, but as it turns out, there is an actual increase in the synthesis of these compounds in many plants. There's currently two theories. The first one is called the photoprotection theory, and the second one is the leaf signaling theory, or the coevolution theory. In the photoprotection theory, the idea is that it's protection against reactive oxygen produced by light, and that the pigments act as antioxidants. Now, the leaves, of course, are exposed to high light intensity, and so it's also thought that perhaps these molecules help to prevent damage of other cellular structures. The leaf signaling theory, or the coevolution theory, is based on the interaction between insect and plants, and perhaps it's a signal to insects that the leaves with the pigments are of lower nutrient value, so not a good place to get food or lay eggs. So the adaptive advantage to the plant is that it will have a decreased parasite load. But the problem is that there seems to be some insects that actually have a preference for leaves that are red. So I guess more experimentation needs to be done. Anywho, I hope that you enjoyed this video lecture, and we'll see you in cell walls. Bye for now.